Hi, I'm Chris Cifelli, and I am the Junior IAC Rep and the Chair of the Science Translation Committee, and I want to welcome you to our next session, which is the IAC SFA Innovation Talks. We have 4 really exciting talks, and I hope you'll be as engaged in this as you have throughout the other parts of the conference. So I'd like to hand it over to Brendan, who will introduce our first speaker, Victoria.
Hello, everyone. Brendan Daisel here, Executive Committee Member for the Students and Fellows Association. It is with great pleasure that I introduce our next speaker, Victoria Onwuliri, who is based in Nigeria and is going to share some of her exciting findings from her graduate work, which focuses on the usage of beneficial effects of probiotic and prebiotic usage on the skin. And take it away, Victoria.
Hello. Okay. Hello, can you hear me, please? Okay. My name is Victoria Onwuliri. Thank you, Brendan, for the introduction. And I will be speaking on the topic, can tropical cream containing Lactobacilli control body malodor? Okay. And I would also like to say that this work, in as much as I'm the speaker of this particular presentation, I have co-authors and supervisors as well, Dr. Kingsley Anukam and Professor Nneka Abakoba. I will talk briefly on skin microbiome. The human skin microbiome is a diverse community of microorganisms that inhabit the skin, ranging from bacteria, viruses, fungi, and even ectoparasites. They help the skin to perform its protective function by supporting the immune system and fending off pathogenic threats. The human skin is the outer layer and a large organ of the human body, and it has different sites. So these microorganisms have different populations residing in different sites of the skin. Sites, those sites are divided into the moist, sebaceous, and the dry sites. The axillary fossa, of which this work is emphasized on, is characterized into the moist site, and we have Streptococcus species, Corynebacterium species, Cutibacterium and Cutibacterium as organisms that are most predominant in the moist site of the skin. Okay, these organisms I earlier mentioned have been associated with body malodor, and they do so by transforming odorless precursors into smelly thioalcohols and sulfonyl alkanols. And alkanols. Sorry for that, please. As adults, we are very conscious of how we smell and how other people perceive us. So this consciousness have made us to understand that body malodor suppresses self-confidence and diminishes social interaction. So this is the most common reason why a lot of us, young adults especially, over the world, all over the world and in Nigeria, tend to use antiperspirants and deodorants in order to obtain an appealing body odor and reduce sweat production in the axillary fossa. Well, various studies have been done, one of which my supervisors and I, we did one that we worked on the effect of antiperspirants and deodorants on the effects of antiperspirants and deodorants on axillary skin microbiome, where organisms that have been associated with body malodor production, likes of Corynebacterium, tend to be higher in people who reported to regular use of deodorants and antiperspirants. From this figure, you can also see that people who reported to use antiperspirants had higher relative abundance of Staphylococcus species. Okay. the use of on one side, cosmetic industries tend to promote the use of antiperspirants. Well, even though these products influence the production of malodor-producing organisms. Then, on the other hand, we know the beneficial effects of probiotics on the gut microbiome and the vaginal microbiome, including the treatment of skin acne. But when it comes to its use in the control of axillary skin malodor, it has pretty much been less studied, and it's not yet in the radar screen of most cosmetic industries. So with this information, we hypothesize that the application of topical cream fortified with Lactiplantibacillus pentosus KCA1 could actually decrease the relative abundance of malodor-producing organisms in the axilla, as well as downregulate genes encoding pyridoxal phosphate-dependent enzymes that are involved in the breakdown of these odorless precursors into volatile and smelly components by these malodor-producing bacteria. We chose this particular probiotic because of its beneficial effects, which are, it's a probiotic of Nigerian origin. It has been proven to produce D-lactic acid and L-lactic acids, biosurfactants, hydrogen peroxides. It has a strong antimicrobial effect, and it inhibits the proliferation of skin pathogens like Cutibacterium acne. It lacks antibiotic resistance and is also safe for human use. One may ask, how did we do it and what did we do to achieve this? We collected axillary skin swabs from a small population of about 25 adult volunteers, consisting of 12 males and 13 females. After the collection, we transferred the swabs into vials containing lysis and DNA stabilization buffer. We now handed over the probiotic fortified oil-based topical cream for them to use twice daily for a period of 14 days. And then we collected the axillary skin swabs. Bacterial DNA was extracted, amplified, and sequenced with an Illumina IMISEC platform. Microbial taxonomy to species level was also generated using Green Genes database on Illumina-based Space Hub. Well, we prepared the cream using natural ingredients like coconut oil, shea butter, and cocoa butter. Reasons are because the participants were really concerned about this adverse effects of this cream. So we wanted to use natural-based ingredients so that we have less or no reports on adverse effects. We also included lavender oil for each participant because of the refreshing, so as they can feel refreshed, since we also told them not to use antiperspirants for the period of 14 days. And then we incorporated the probiotics into about 12 grams of the Lactiplantibacillus pentosus KCA1 into the oil-based probiotic cream. That's how we formed our cream, our topical cream, and handed it over to the participants to use for the period of time. Well, after the 14-day use, of the KCA1 probiotic cream, we observed that it had a great impact on both male and female axillary skin microbiome. There was a great, at the filler level in females, we observed increase in the relative abundance of actinobacteria and proteobacteria, and surprisingly, a decrease in the relative abundance of actinobacteria and proteobacteria. Surprisingly, an increase in the relative abundance of firmicutes. Same effects was noticed also amongst the male participants. Through this observation, we are suggesting that the topical cream really had an effect on the female axillary skin microbiome when compared to that of the males. At the general level, likes of Corynebacterium and Cutibacterium that has been implicated in malodor production were strikingly decreased. We are slightly decreased with Staphylococcus and likes of Lactobacillus on a very high side. Same effect was seen on the male participants after the application of the cream. Well, at species level, likes of Corynebacterium tuberculostericum that has been implicated also in the production of axillary skin and production of body malodor was seen to be decreased after the use of the cream, followed by Corynebacterium reageli, appendices, and genitalium. There was also an increase in the Lactobacillus species diversity. For males, the number of species increased from 29 to 49 species, and for females, from 26 to 38 species. Same also was observed amongst the male participants in case of Staphylococcus hemolyticus, Staphylococcus epidermidis. These are also species that have been implicated in production of body malodor. So the effect of this topical cream after 14 days was actually seen in the reduction of the relative abundance of some of these malodor producing bacteria. Okay, we also observed that the PICROS results generated for bacterial metabolic functional genes involved in the biotransformation of malodor precursors to volatile thiol alcohols was downregulated, likes of pyridoxal protein, PLPDE protein, and pyridoxal 5-phosphate synthase. These enzymes were downregulated after the 14-day application of the KCA1 cream, both in males and in females. Well, in conclusion, there was a significant decrease in the relative abundance of malodor-producing Corynebacterium and Staphylococcus species, both in male and female. There was also an increase in the species diversity and abundance of Lactobacillus, which is a non-odor-producing organism, by over 385%. On the other hand, we also saw a downregulation in the production of pyridoxal protein-dependent enzymes that are involved in the malodor production by these malodor-producing organisms. It was very interesting to get positive feedbacks from the participants after the 14-day use of the KCA1 probiotic cream. All the participants gave positive feedback. There was no skin reaction, no adverse reaction, no armpit odor. In fact, one of the female participants really gave a thumbs up after the 14-day use. She complained seriously of having excessive sweat production and armpit odor, and she never experienced those within the 14-day use of the probiotic cream. I wish to sincerely acknowledge the participants who consented into participating in this study, and I will not leave my supervisors and mentors out of this, because they were the ones that motivated me to be a partaker of this study. And this study was funded by the UBiome that granted my supervisor, Dr. Kingsley Anukam, a grant-in-kind award in 2018. And my supervisor, Professor Nneka Agbakoba, who motivated me to be a part of this study. So thank you very much for listening, and I appreciate questions that would come afterwards.
Awesome, great talk. So we're running out of time here. I guess just to for the sake of time, I'll just ask you kind of one question or bring it up. You know, the corny bacteria are in the gut at least cause a large inflammatory response. So it's interesting to consider how topical probiotics might be used for anti-aging effects and things like that. Are you looking into anything in that in post work?
Yes. I think this work needs to go beyond this, because owing to the fact that we use a smaller population, I think if we use a larger population, we could get a very promising result, because with this population alone, we got good results. So I believe with the larger population, we will get more promising results.
Awesome. All right. Thank you for the great talk.